Good afternoon. I'm Patricia Sampuri, and I will be serving as your co-moderator for this session of the e-Symposium for PRISM. My co-moderator is Rachel Friedman, who's working behind the scenes to move our slides along. Rachel, please say hi to everyone.
Hi, everyone. Thank you so much for being here.
You are on the technology concurrent session, which will run from 5:30 to 6 p.m. In this session, our students will be presenting their research, which is related to topics on computer science and software development. During a person's presentation, if you have a question, please type it into the Q&A, hit the Q&A button and then type in your question and submit it. And I will look through them at the end of the sessions, and time permitting, we will ask at least one question. Okay, let's get started. Our first presenter is William Higgins. Today, William will be talking to us about finding out what makes us human through bioinformatics. Take it away, William.
Hi, thank you so much. So what is one thing that we all have in common? In fact, we're all human. One of the first questions going through my head, although not the main question of the research, was why we are the way that we are. When starting both my classes here at John Jay and the research with my two mentors, Dr. Nathan Lents and Dr. Hunter Johnson, I've slowly come to a greater understanding of my first question. With one solution comes another problem, because when starting my research, I was presented with a whole new topic to think about and question, which was the origin of humans and orphan genes. Orphan genes are a gene script within the DNA that is specific to the species or species lineage. With this topic, my mentors presented me with a question to solve, and that was, where are these orphan genes located and how do they relate to chimps? To solve this, I had to, and I still am, having to overcome an obstacle, which is learning computer programming. Although I am way better than when I started, I'm far from being a master. To be productive while also learning a whole new language can be difficult at times, but there are a couple tools that move my experience forward. Python being one of them was the coding language that helped me connect to all the tools in order to help locate orphans. Next slide, please. Before we could tackle the human genome to find what we're looking for, the search needs to become more narrower, so it would take the least amount of time possible. That is when one of my mentors, Dr. Nathan Lents, hypothesized that looking for orphan genes on microRNAs would be able to show that the genes will end up on some of the best possible candidates. What is microRNA, though? MicroRNA is a specific form of RNA that mainly focuses on regulation of gene expression. So now that we have a general location of these orphans, we need a method of finding them using Python. Next slide, please. Luckily, I have some awesome opportunity to work with Dr. Johnson to learn how to use Python and how I could use it to manipulate the human genome. Two major tools that were being used were BioPython and online databases. BioPython is a program within the Python that helps align genes by having the ability to be upgraded through the various online packages. Why is this useful? We use these alignments from, let's just say, chimp and a human to determine what is unique among them. In this photo, we have PyEnsemble. PyEnsemble is one of those online packages in BioPython that allows you to see all known genes based off the website Ensemble database. This was a great learning tool for me because I was able to play around with the genes and code that I never even knew existed, like the gene's ID and the various type of genes, one of them being microRNA. Next slide, please. Some of the stuff I also got to play with, like I said before, genes and codes, but I also got to stuff that seemed a little more tied to bioinformatics as well. For instance, this photo shows early works of my Python code when dealing with genomic alignments. Note the sequences that are in red were mostly made up on the spot because I needed to know that they would specifically align. And when dealing with random stuff, I don't want to see stuff that doesn't work when learning, of course. Next slide, please. So where do we get all this information to sort? Thankfully, there are multiple online databases, like I said before, Ensemble, and there's also ones called MirrorBase and NCBI that have all these genetic information available to the public in easy-to-use FASTA file format. FASTA files can be manipulated and sorted, which is the best possible when dealing with human genomes, similar to the files dealt with using PyEnsemble from the two previous slides ago. But except those were in GTF or gene transfer format. Next slide, please. As can be seen here for the results, they were successful alignments between the chimp, which is the bottom sequence, and the human, which is the top of the microRNA. These three aren't the only ones on the list, since there are nearly 600 just like it. With these alignments, finding the evolutionary track of these genes will come shortly. The scores shown help visualize how well the chimp and the human microRNA align. Unfortunately, the scores here aren't quite useful yet because they're heavily affected by the sequence's length, which is thankfully brought to my attention by Dr. Johnson. Next slide, please. As can be seen with the results, our work is far from over, and we still have so much to uncover about these orphan genes. I will continue to look at these alignments as well as create newer and more accurate ones. In the future, once this is finished, I see this spreading to many different fields as well. Next slide, please. For instance, the research holds so much information about gene regulation and expression, which is key to unlocking the solution to my last problem, cancer. According to the World Health Organization, in 2018, approximately 18 million people have been diagnosed with some form of cancer. I want to use the understanding of genetic origin expression with this research to help my future and everyone else's future as well. And next slide, please. I would like to thank my fellow lab mates, Beatriz, Jessica, Jose, and Samantha.
Thank you very much, William. And as William's program presentation shows, he actually did a combination of using technology as encoding with biology. So that's why he has two mentors, Dr. Johnson and Dr. Lents. We do have a question for you, William. Someone would like to know, why do you hypothesize that microRNAs have a high abundance of orphan genes?
Well, although it's still open to debate, the microRNA has a very big role in both gene birth and gene death, which is, I believe, well, gene birth being and gene death being what they sound alike, of course. Gene birth being kind of like the creation or birth of a new function of a gene, and gene death being something like the gene shutting down. And since these genes are now new to the organism or how it's or the organism's lineage, we now have an orphan gene, which because it's uniqueness.
Okay. Okay, very interesting. Interesting presentation, and thank you very much.
Thank you.
Our next session is actually going to be given jointly by Zafar Seenauth and Hunter Kearney. Before they start, just a quick reminder about the Q&A button at the bottom. So if you do have a question for either Zafar or Hunter, please type it up, submit it, and time permitting, I will ask the question. These gentlemen will be talking to us today on how does malware behave in a virtual environment. Take it away.
Hi, everyone. Before we get into how they behave, first we must understand what a virtual machine is. Virtual machines are like a computer inside a computer. A guest operating system, OS for short, is installed within the host OS. The OS running in the virtual machine is kept isolated from the host OS. Therefore, malware running in the virtual machine cannot harm the host OS. If the malware damages the virtual machine, you can reinstall the OS in the virtual machine or return the virtual machine to a clean state. Next slide, please. On the left, we have some examples of commonly used operating systems, and on the right, there are some examples of virtualization. With this in mind, we can try to look at how malware itself behaves inside of a virtual machine. We believe that there will be a vulnerability that can be exposed or exploited by malware. Our hypothesis was basic to the point of whether or not malware understands if it is within a host OS or a guest OS. If so, then we can suggest that virtual machines are not truly safe environments for testing. And on the other hand, if the malware does not recognize that it is within a guest OS, then we can suggest that all testing should be done within a virtual machine. Next slide, please.
The main research approach taken is static and dynamic analysis of the malware within a virtual machine. Static analysis refers to the process of analyzing the code or structure of the malware to determine its function. The program itself is not run at this time. The image on the right is an example of a static analysis using a program called Dependency Walker on a malicious program. This revealed the function winexec, which tells us that the malware will execute another file on the disk somewhere. Next slide, please. In contrast, when performing dynamic analysis, the malware itself is ran and then analyzed. This program, Process Monitor, is commonly used during dynamic analysis as it captures all processes that are ran by the malware. In this image, the malware in question is shown to create files, modify registry keys, and then save and close all on its own. Next slide, please. The expected results of testing the malware within the virtual environment is to be that vulnerabilities would be found. When analyzing changes within the program, there's an expectation that malware would either detect it being within a virtual machine or escape from the virtual environment. This is our expectation because as of recent times, there has been an increase in malware that has been able to find exploits within a virtual machine. Given the time frame, more malware can be analyzed to find exactly where within the machine assembly language the malware resides so that threats are better detected. This has to be done within a separate machine to isolate possibilities of malware leaking into the host OS, which we tested using a Mac Pro running VirtualBox and VMware. Within these environments, we use simulated malware with free open source tools to test whether or not we would find these results. Next slide, please.
To conclude, the main point of this research is to present potential vulnerabilities in virtual machines. Virtualized software, such as virtual machines, are supposed to be secure environments, so finding potential vulnerabilities can be insightful in regards to mitigating and preventing any zero-day exploits or any other underlying issues within virtualization. The information collected from this research can help a wide variety of users and organizations since virtual machines are applied to several computing paradigms. If malware can escape the virtual machine and achieve privilege escalation in the host operating system, then this is an issue that won't resolve on its own. However, finding out how malware can escape the virtual environment can lead to mitigation and preventive measures for future exploitation. It is exciting to possibly find vulnerabilities in a security utility like virtual machines, since so many information security experts and organizations use virtual machines. This research has the potential of establishing groundbreaking opportunities for the information security industry. How will the discovery of vulnerabilities in virtualized software impact other information security practices? There's not a simple answer to this besides to prepare like any information security expert should in the event of vulnerabilities like zero-day exploits. In general, findings of vulnerabilities in virtual machines can lead to new ways to detect malware, which depends on what is actually found in this research and coming up with new ways to combat security threats as a whole.
Virtual machines aren't perfectly developed pieces of software. With this research, finding out how malware may escape the secure environment of virtual machines is the ultimate goal. The significance of this research is to expose the flaws that are present in virtualization software and address the potential exploitation that could put those that operate within virtualization at risk.
Okay, gentlemen, I hate to cut you off, but you have used your time up for the time being. Well, for the session. I'm going to ask you, though, to hang out a little bit. And if we have some extra time at the end, I do have a question that popped in, so we will ask you that as well. So thank you very much for your time. And now we're going to move on to our next presenter, who is Beatriz Chantel Mercado. Beatriz is going to talk about the discovery and classification of a unique microRNA. And like our previous speaker, William, she has two mentors, one on the technology side and one on the biology side. And before I turn this over to Beatriz, one more quick reminder, if you have a question for a presenter as you're listening to them speak, please click on the Q&A button, type up the question, and time permitting, we will ask them the question. Okay, take it away, Beatriz.
Okay, thank you so much, Pat. So, next slide. During this research, I have focused on identifying and characterizing regions of the human genome that have played a key role in human evolution from our primate ancestors. The genome of all apes have been sequenced, allowing for further research into the evolution of human beings. For reasons I will soon explain, I have focused specifically on genes for microRNA molecules. MicroRNAs are post-transcriptional regulators that bind to specific targets, messenger RNA, leading to a silencing of gene transcripts. I searched for microRNAs that are taxonomically restricted to humans and thus have no orthologs in other species. Next slide. It is known that the human and chimpanzee lineage split from each other 6 to 8 million years ago. Also, these species have diverged a remarkable amount in terms of anatomy, cognition, and behavior, despite sharing nearly all the same protein coding genes. One hypothesis holds that the differences in gene expression holds the key to human uniqueness. Next slide. Humans have about 20,000 protein coding genes, and nearly all are shared with our closest living relative, the other African apes. We also harbor over 40,000 RNA coding genes, and about 2,000 of those code for microRNAs. Most microRNA genes are shared closely amongst related species. However, there are species-specific microRNAs, and their origins remain a mystery. Next slide. To find human-specific microRNAs, we use a computational tool called MUMR4 algorithm for genomic alignment, which identify tracks of similarity and dissimilarity amongst two sequences to a given genome. We began our search on chromosome 21, scanning for microRNAs that aren't found in the chimpanzee genome. This reveals several microRNAs that are clustered in families of 2, 3, or 4 members that are identical or nearly so. This led us to hypothesizing that recent segmental duplication occurred on chromosome 21. I selected miR6724 genes with 4 members to begin my analysis. Next slide. Firstly, I performed comparative sequence alignment of miR6724 for family members. This revealed that they all have the same exact mature sequence, which is highlighted in yellow. If expressed, this microRNA family targets the same messenger RNA, making these 4 microRNA genes functionally redundant. There are no known chimpanzee orthologs for miR6724. So I searched for orthologs using a technique called black search, which is a simple search algorithm for detecting closely related sequences. Next slide. miR6724 mature sequence was found in the other great apes as well. However, there has been a substantial divergence, particularly in the human sequence, indicating rapid evolution. Using a program called RNA-fold, I generated the predicted structure of the microRNAs as well. While there is substantial structural conservation among the apes, the sequences divergence is substantial. Next slide. We then set out to look at chromosome 21 as a whole. And we noticed that there's a distinct cluster of microRNA families. These microRNAs are highlighted with arrows, 2 of which are human-specific, miR6724 indicated with a yellow arrow, and miR3648 with a red arrow. Additionally, there are many ribosomal RNA genes within this cluster as well. Next slide, please. These human-specific microRNAs are related to a single sequence in other apes, suggesting recent duplication events in the human lineage. We generated a synteny map to detect recent duplication by comparing chromosome 21 to itself. Additionally, we use LD split to search for recombination hotspots, which increases the frequency of mitotic recombination. This revealed a distinct cluster of recombination hotspots near the cluster of human-specific microRNAs and the ribosomal RNAs within chromosome 21. Next slide. Although it is impossible to definitely prove exactly how molecular events occurred millions of years ago, our data provides strong evidence for a working hypothesis. Our hypothesis is that multiple new microRNA genes were formed in an ancestral population several million years ago through segmental duplication occurring within an array of ribosomal RNA genes. Due to microRNAs being small in size and transcription silencing nature, these microRNA genes evolve rapidly and contribute to the evolution of our lineage. Next slide. I would like to thank PRISM and the Lents Johnson Lab for making this research possible.
Thank you very much, Beatriz. Another very interesting presentation, a lot of information in there. And we do have a question for you. Someone would like to know what your next steps in research will be.
Okay, our next steps is to analyze the predicted targets of these microRNA genes. And so far we have several microRNA genes that are associated with embryonic development of the nervous system. So we are suggesting that the segmental duplications that occurred on chromosome 21 also play a role in human evolution, specifically in the human brain.
Very interesting. Sounds like you'll be very busy when you get back to campus. Thank you, Beatriz. All right. Our next and final presentation for this session will be by Joseph Brown. And Joseph is going to be talking about a hybrid dynamic authentication scheme for multi-factor verification. Once again, if you have questions, for Joseph, please type them up in the Q&A section and submit them, and time permitting, we will ask him your questions. All right, Joseph, it's all yours. Go ahead.
Great, thank you. Next slide, please. So this is a quick overview of what's on our agenda to talk about today. Next slide. So one of the biggest issues in modern cybersecurity is a lack of improving authentication norms. When you log into a website, you're generally greeted with a pretty well-accepted interface, that being a username and a password. Logins typically work through hash comparisons, which means a single input is compared with a verifiably equivalent output. But given threats like brute forcing, a lot of people don't know how insecure these operations really are. It's pretty well agreed upon they're insecure, but if they're so outdated, you might ask, why don't we just adopt new methodologies? Surely other options exist, and the answer is yes, well, they do. However, many of these are not holistically secure. One example being two-factor authentication, which is typically floated as an alternative, being proved insecure in a 2016 NIST study. Two-factor authentication and most other alternatives tend to be insecure because they simply pad the complexity of the client and server, thus leaving the transactions between them as insecure. Next slide, please. So this is where we started to think, how could we protect the data between the client and server instead of just artificially padding either end? We surveyed the field for authentication proposals, as well as studied current parallel methodologies used to secure communication, such as SSL. We came to a pretty simple conclusion. Any improved authentication methodology must include these points. Multi-factor, as in verification isn't just done through a single validation, but according to a multiplicity of factors. Dynamic, in that data sent between the client and server should not be reusable between sessions. And flexible, in that the scheme must be able to be adapted alongside other improvements. That is, say, if our scheme is focused on preventing man-in-the-middle attacks, then it must be flexible enough to integrate with other proposals making separate improvements. Next slide, please. That brings us to our research approach, or how we went about developing a scheme based on our hypothesis. A successful scheme should be able to integrate all of these standards listed on this graph, which are essentially derivatives of the points in the hypothesis and color-coded to the same scheme, along with the more generalized self-evident evaluations, which are pointed out in purple. These include factors like speed and simplicity. Next slide, please. So this brings us to what we actually developed given this information, which is a scheme we dubbed the two-factor hash verification. We made this scheme by abiding by extrapolations of the factors we had determined prior, which we had discussed in the hypothesis slides. So to summarize how this works, instead of simply giving a key into a door, imagine you had a key split into two halves. The client has a half and the server has a half. But instead of sending the key half, you're sending the blueprint for said key half, which will be reconstructed with the other half on the other end, thus making the full key. The blueprint changes slightly each time you log in, but is still derivative of the other key scheme. Thus, only two people ever have these respective halves, the client and the server. The halves are a derivative of data, which is itself derived from an input which is in usage similar to a password. Thus, as long as you can produce from the front end something which creates one input and output, even though this input is never sent to the other end, it can still be integrated into this system. Next slide, please. So we did both a quantitative and qualitative evaluation of what we created, as we had implemented it in Python instead of simply keeping it as a theory. First and most importantly, it worked with a simple client and server, so that it didn't involve any extra nodes or anything of the sort. We ran time complexity tests for brute forcing and found that although it was not focused on brute forcing, it did actually provide stronger brute forcing protections in the current authentication paradigm of hash comparison logins, so no extra weaknesses were found. It did run slightly slower than current logins, but only by about a quarter of a second maximum. Our evaluation showed that it was also holistically secure against interception and impersonation attacks, provided protections against attacks such as sniffing, man-in-the-middle, masquerade, and replay attacks, for example. Next slide, please. That leads us lastly to future discussions. Now, we mainly thought it'd be a good idea to focus on a few things in the future: integration with two-factor authentication, which we didn't get time to check, ideally for maximal anti-brute forcing protections, and then also improving a one-time vulnerability in registration, which was the only time there was a risk, although this risk is visible in all authentication systems in general. Lastly, we only compared this with two other studies and of course the current paradigm as well. So in the future, we'd like to compare it with more studies for brute forcing and time evaluation comparisons. Next slide. And that's all for now. I'd like to thank the John Jay Honors Program and also Professor Obaidat for mentorship.
Great. Thank you very much, Joseph. We have a question. Why did you need a different I can't speak today, authentication scheme? Don't large companies already use 128-bit encryption?
So the issue was less with the encryption standards and more with the typical standards of schemes in how they're applied to users rather than how they're used in a database. Typically because in any system, it's not specifically the company, which is typically at risk, it's the individual user. So in the case of logins, for example, a lot of times passwords are compromised, people ignore responsibility surrounding password security, or alternatively, they fall prey to phishing, or they fall prey to, like, say, fake SSL attacks. So because of that, we wanted to develop a scheme which was more holistically secure for a user as opposed to storage in a database.
Well, I feel safer. Actually, we're at 6 o'clock, but I do want to get a question out to Zafar and Hunter. You had 3 questions, but I'm gonna bring it down to 1. Someone in our audience wanted to know, can hackers develop a cyber weapon to infect your host OS? I'll let you guys decide who's going to answer. Hunter, Zafar, you still with us?
I'm sorry, hello? I'm sorry.
That's
what
okay.
was the question again? I'm sorry.
Can a hacker develop a cyber weapon to infect your host OS?
Yes, not necessarily a weapon, but there's a malware known as VM exit that's been used as a skeleton for new malware that can escape virtual machines. And
Okay.
yeah.
Okay, so it can happen.
Yes.
All right. Well, with that, we conclude this session. I'd like to thank all of our presenters for their hard work, and I'd like the audience members to please give everyone a virtual round of applause. Our plenary session with our keynote speaker, Dr. Olivia Orta, will be starting next. Dr. Orta will discuss her career trajectory from a John Jay student to her time at Harvard University while sharing lessons she learned along the way. To join the plenary session, you need to go back to the website, www.jjay.cuny.edu/PRISM, that's PRISM as in Mary, and follow that link at the bottom to join the second plenary session. Thank you to all of our presenters and our audience members. Enjoy the rest of the symposium. Goodbye.